All right, so let me go ahead and maximize the size of the screen, and let me show you how this is going to work. So this is going to be an experiment that you're going to do online. The experiment is going to be conducted on a plant. This is a water plant, and it's inside of a container that's holding the water. You can see the water level right here. It has light. There's a light bulb up here, and you can see the cord coming out. And it has a meter to check for oxygen. It has a meter that checks for temperature. The light right here is measured in intensity. That tells us how much light there is. And then it also tells us what the CO2 level is. So if we remember about photosynthesis, there are some different things, and I'm going to show you this. So on your task card, we have our model for photosynthesis right here. This is that chemical formula that tells us what happens. And so on the left side of the arrow, we see CO2 and water and light energy. And then on the right side of the arrow, we see glucose or sugar and oxygen. So the items that are on the left side of that arrow, that's the CO2 and water, those are the reactants, and you should have this in your notes from yesterday. And then on the right side of the arrow, we have sugar and oxygen, and those are the products. Did someone have a question? What do you mean the water intensity? On this sheet? I was talking about this equation for photosynthesis right here. Yeah. And so what you're going to be doing is you're going to be looking at this experiment and looking at the different, what we call variables of photosynthesis. And variables are things that you can change in an experiment. And so in this particular experiment, you can change 3 things. What are those? Which ones have bars that I can change things on? I can tell you one of them. It's temperature. I can change the temperature by adjusting the bar. See, when I hover over it, I get a little hand. What do you guys think the other 2 are? All right, perfect. So we've got somebody who's finally volunteering here. So we've got light and we've got CO2. So our light intensity we can change and our CO2 we can change. Perfect. All right. Now for this gizmo, we have to change those variables, those things that we can adjust or vary to answer the questions. And so when you have your task card up, look at the investigation number 1, which is the second page. If you printed it out, it's the second page digitally, but you have to scroll down to get to it. And step 1 through 4 is just setting up this gizmo. So you're pulling it up. You're going to switch over on the right side of the gizmo to a bar chart. So you'll see some tabs along the top, and you're going to click on bar chart. And then you also need to click on show numerical values. So you click on that, and that gives us some data in the form of numbers. And then we need to continue to read on through the tasks sheet, and it tells you what your next steps are going to be. The prediction is the main part that I wanted to talk with you guys about and make sure I made a recording. So when you get past reading about the background, you get to a part called predict. And this is where you're going to start filling in some questions and answers kind of thing. So in the prediction, it says, what conditions do you think will maximize the rate of photosynthesis? And the way we know the rate of photosynthesis is maximized, we get the most oxygen showing up on the bar chart right here and on this gauge right here. So you have to move these variables around and see which ones cause us to get the most oxygen. And on your chart, it says ideal for the spot that has the most oxygen. So what we're going to do is I want to do the very first one with you. So we're going to start with temperature, and I want you to do this at home along with me. The first part says predict. So we're going to predict which temperature is going to give us the most oxygen. Do you think the temperature going up or going down is going to give us the most oxygen? Up. Okay, so pick a number. So this is starting at 30 degrees Celsius. You can use your mouse to move that bar up. So pick what number you want. Do you want it to be all the way up at 50? Do you want it to be 42? Where do you want it to be? And you pick and decide where you want that number to be. And write that in your prediction box, or type it, sorry, which is right here. And then once you have written that down or typed that in, move your temperature bar to what you said you predict is going to give you the best number. I'm going to predict 50. So I'm going to lift mine all the way up to 50. And then I'm going to look over here and see what happened with my oxygen. Did it give me the max? Mine, it didn't. Mine didn't at all. That was not a good choice for me. So I need to look for the ideal, the actual amount that's going to give me the most oxygen. So I need to go back over here and grab my bar for temperature and raise and lower it until that oxygen value goes up to the highest it can go. Did anybody do that yet? I'd like to hear what you got. Type in chat, please. What numbers are you guys getting? Come on. 24, lots of 24s. Okay. Yeah. Okay. Yeah, there's a bit of a range, right? All right. So, yeah, 24, more 24s. Yeah, there is a range. Good job. That's excellent that you put that range in there. Love it. So the rest of the questions are going to follow along the same route. You'll just leave the temperature where it's at and then move to the next variable, which in this case is light intensity. So you'll make a guess and then see where it actually is ideal. Okay. And I'm going to go ahead and stop my recording here. For those of you who are watching this video for instruction, you'll want to go ahead and continue through the rest of this and answer the questions down here, these reflection questions that you're going to complete to finish your lab. And that is it for today. Hold on, guys.